Hello students, in the last two videos of Marcantia, we have studied about the external morphology and internal anatomy of the Marcantia. In this session, we'll be studying about the reproduction of Marcantia. How Marcantia reproduces. Marcantia reproduces by two methods, that is, vegetative method and sexual method. You know what is sexual method? It is the formation of new plant from the zygote. That is, it involves the formation of gametes. It is haploid in nature. The haploid gametes are the antheridia and archegonia. The antheridia is the male gamete and the archegonia is the female gamete. When it fuses together through the process called as fertilization, they form the diploid zygote, and this diploid zygote develops into a new plant. So this is about the sexual reproduction. But in this session, we'll be concentrating on the vegetative part of reproduction. The vegetative reproduction involves the development of new plants from the diploid cell itself. It do not involve the formation of gametes or the fusion and fertilization is all not involved. It involves the formation of new cells from the somatic cells. This vegetative reproduction involves three different methods. Three different methods of vegetative reproduction are: the first method is reproduction from the separated thalli or the dead and decaying thalluses. Okay. The second part of reproduction is, second type of vegetative reproduction is development of adventitious branching. And the third type of vegetative reproduction is gemma cup or gemma formation, gemma cups or gemma formations. Okay, so this is the vegetative reproduction. We'll study in detail about the vegetative reproduction. Okay, moving forward with this. Coming on to the first type of vegetative reproduction, that is the progressive death and decay or development of new cells from the fragments of the thalluses. So what is this progressive death and decay? When you consider the thallus of the Marcantia, this is a thallus, two-dimensional structure of the thallus, which is strong. When you consider the thallus of the Marcantia, it is flat and dorsiventral in nature, and it reproduces or it undergoes dichotomous branching at the apical end, whereas the posterior end has the older cells. So when you take the Marcantia's thallus, the thallus has the anterior end, which is the growing end, because it has the notch-like structures, which helps in the development of dichotomous branching. And it has a posterior end. This posterior end has the older cells. Newer cells are found in the anterior end, whereas posterior end is made up of the older cells. How long the older cells will be? You know, every cell has its own life cycle, and that is period of okay. So it has every cell has its own lifespan. So here, even the older cell also has its own lifespan. As the new cell divides and grows, the older cells get age and they undergo deformation, death. Okay, they undergo death. When the older cells die, okay, when the older cell die and they get decay, the newer cell, okay, newer part of the thallus or the branching part of the thallus, since it is dichotomously branched, the branch gets separated from the older parts, and each branch of the thallus grows into new plant. How many ever lobes are, so many branches get separated and new plants are developed. So this is how the older part dies and the newer part develops into a new thallus. So this is how the vegetative reproduction occurs in the first method. Hope you understand. So thallus has the older cells and the newer cells. Newer cells are always found on the apical end, and the older cells are found in the posterior end. As the older cells dies, okay, and decays, the newer cell which undergoes dichotomous branching because of the presence of the notch, okay. So this dichotomously branching part of the thallus will develop into new plant body. From one thallus, here in the second picture you can see two thalluses are developed. In the third picture you can see 1, 2, 3, 4 thalluses are produced. So 4 fragments of the dichotomous branching develops into 4 different plants, 4 new plants. So this is how the older one thallus are developed into many new thallus, new plant bodies. Okay, so this is the first method of vegetative reproduction. The second method of vegetative reproduction includes, okay, the development of advantageous branching. The development of advantageous branching. What is this advantageous branching? This is a special type of branching. You know, branching occurs at the apical ends of the thallus where the notch is present. Okay, whereas advantageous branchings are the branches which develops from the lower part of the thallus. Okay, that is ventral part of the thallus, the part where the thallus or the plant faces downwards, from where the new branching arises. This new branching which arises from the lower part or the ventral region of the thallus is called as advantageous branching. These advantageous branchings get separated because of various activities. So this separated advantageous branching develops into a new plant. Okay, so this is the second type of vegetative reproduction. Moving further, the third type of vegetative reproduction. The third type of vegetative reproduction is by formation of gemma. This is very important, okay, for the exam point of view. They will be asking you the development of gemma in the vegetative reproduction, okay, for 5 marks with the diagram. And even in the practicals, you'll be getting gemma, the TS of the gemma, okay, so that you have to draw and explain the characteristics. So when you consider the gemma is developed into the cup-like structure called as gemma cups. Okay, if you remember the external morphology of thallus, the external morphology of this thallus, okay, shows the thalluses are dorsiventrally flat. Okay, the thalluses are dorsiventrally flat and they have the midrib region. I told on the dorsal region, on the upper region of the thallus, they have divided into, okay, different parts with the help of midribs, and the cells here and the thallus is also rhomboidal in shape on which the pores are present. Midrib is present. Okay, so on this midrib they develop a cup-like structure. It is nothing but it is a same tissue, but okay, of the thallus which develops into cup-like structures. Even this cup-like structures has different parts. It is same as the VS of the thallus. So when you take the cup-like structure under the microscope and view, okay, the cup-like structure which developed is called as the gemma cup. The gemma cup is also made up of 3 layers, as that of the internal anatomy of the thallus. That is the apical region. The what is it? Apical cells. Okay, the upper region and lower epidermis. So upper epidermis and lower epidermis, and it has the air chambers, that is the photosynthetic region, and also it has a storage region. Okay, but the shape is here, it is in the cup-like structures. What is the size of this gemma cup? Gemma cup is around 2 millimeters to 3 millimeters, 2 millimeters into 3 millimeters in its size, and they are encapsulated, okay, and they are present on the dorsal region. As I told you, these gemma cups are found on the dorsal region, and these, okay, are developed in the midrib regions of the thallus. And this gemma cup has, okay, develops a stalked structure, small stalked structure, which is bearing a dumbbell-like structures. Okay, which bears a dumbbell-like structure. Okay, this stalk-like structure forms the gemma proper. Gemma proper is the dumbbell-like structure, is called as gemma proper. The stalks are found onto the apical region, which is found inside the cup. Okay, and this gemma proper has many cells. It is multicellular in nature, and these cells are usually parenchymatous, and they contain the chlorophyll pigments. So these gemmas are also these. This is the gemma. This is the gemma which is found inside the gemma cups, which develops on the interior layer of the gemma cup. Okay, from the interior layer they develop the stalk-like structure. The stalk is thin, and on the stalk you find the biconvex shaped structure called as gemma. This gemma is green in color because of the presence of chlorophyll pigments, and they are made up of number of cells. And in between, they are also made up of the oil cells and the mucilaginous cells, which stores the oils and mucilaginous substances, which is formed during the photosynthesis. And this gemmas are biconvex shaped, okay, convex shaped, or it is dumbbell shaped. Why? Because this gemma is not spherical. In between the gemma, you can find a constricted region, region where it is formed, it is curved inwards. It is a constricted region here present. This constricted region has number of cells, okay, and it has a thickening, okay, because of number of cells which is present. It is thick in nature, okay. So thick region is present because of the presence of the number of cells. This region is called as the notch region. This notch region is responsible for the development of the rhizoids. These are the cells which develops the rhizoids. Okay. Now, when the water is more, okay, due to the water, okay, when the Marcantia grows in the watery areas, okay, these gemmas, okay, comes out of the gemma cups, and they float into the water and fall onto the surface. Okay, so this gemma, when it falls onto the surface of the ground, what happens? Okay, these gemmas, when it comes in contact with the land surface, what happens? This notch region develops the root-like structures or the rhizoids. These rhizoids attaches firmly onto the surface of the land, and they develop into a new thallophytic plant body. This is about the gemma and gemma cup. So this completes the vegetative reproduction. Vegetative reproduction is asexual reproduction, where reproduction occurs by the somatic cells. Based on this, the vegetative reproduction of three types. That is, one is from the dead and decaying cells, where the older cells die, and the newer cells which is dividing at the notch region, which undergoes dichotomous branchings. Each branching separates from the old thallus, and each branch develops into new plant. Second one is development of advantageous branching. The advantageous branching, a special type of branching which develops from the ventral region of Marcantia, and this advantageous branching undergoes fragmentation or separates from the thallus and develops into new plants. Third is by the gemma cup formation. The gemma cups are the vegetative part which is found on the dorsal region in the midrib of thallus. This gemma cup has number of dumbbell shaped or convex shaped structures, okay, which has small stalk on the interior part of the gemma cup, and this biconvex shaped gemma helps in the vegetative reproduction. Okay, this gemmas are green photosynthetic in nature. They are biconvex because they have central constriction. In the constricted region, there are a number of cells which thickens the gemma. Okay, so this is called as gemma notch. From the gemma notch, they have number of cells which develops into the rhizoids. When the gemma disperses from the gemma cup through the water or the other processes, these gemma, when it falls onto the surface of the land, when the gemma comes in contact with the land surface, these develop the rhizoids and develop into a new plant. So this is about the vegetative reproduction of the Marcantia. Next is about the sexual reproduction. Okay, so sexual reproduction will be studying in the next session. Thank you.